The first we have on the program is Martin Lema. Martin, are you with us? Maybe the tech.
Hi, Peter. Yes, I am.
Great, great. Good to see you as always, Martin. And just by a quick way of introduction, Martin, and you can correct me if I'm wrong. You're now in the academic world, but you were for a good period during the development of the Argentinian current policy on genome editing. You were working in the administration, if I've got this correctly. So that's your background. And thank you so much for making the presentation today.
Okay, so, thank you, Peter. Hello, Erica. Hello, everyone. Yes, Peter, I can confirm that I was chairing the Argentine Biosafety Commission on the time that we developed our policy for genome-edited organisms for agriculture, which I'm about to describe now.
Thank you.
Mr. Lema, you can take the control of your slides by clicking.
Thank you. Okay, first of all, in order to develop our policy for new breeding techniques, including genome editing in agriculture, we started from recognizing that we...
Mr. Lema, I'm sorry, you did not take the control of the slides. Please click in the... Okay, please. Now it's working. Yeah.
Thank you. So as I was saying, we, Argentina, as most other countries in the world, have two established regulatory options for new genetic variants. On one side, for new varieties or breeds, including those obtained by mutagenesis, different kinds of mutagenesis. We have one regulator and one set of rules, and then we have another regulator and another set of rules for what we, for now, I will call transgenic organisms. We recognize that sometimes very similar products can be obtained by one or the other route. For instance, sorry, I'm trying to change the slide here. Here we have an example of a drought-tolerant crop obtained by conventional or traditional mutagenesis, if you want to call it that way, used in a different country, in Zambia, or we can have also a drought-resistant wheat, in this case developed by an Argentine biotech company and approved for commercialization in Argentina. So we started by recognizing that those two regulations exist, but now the problem with genome editing is that it created a question if the products belong to one box or the other. So as a brief introduction, our regulatory framework for modern biotechnology, let's call it for now, has been working for 3 decades now. We have over 60 genetically modified organisms, crops and recombinant live vaccines approved for full use in Argentine agriculture. And our regulatory commission is recognized worldwide as a reference in GMO biosafety. Particularly, the criteria for gene editing in agriculture that I'm about to describe is a case-by-case sorting mechanism to decide if each product is a GMO or not. It's based on the Cartagena Protocol definition for LMOs, or living modified organisms. This is the most worldwide recognized definition in this area, and it's the same as the Argentine definition for genetically modified organisms. The decision regarding if it is a GMO or not is made in 60 days. And if the product is a GMO, it has to be regulated as a GMO. But if not, it is under the regulatory framework for conventional products. As I was saying, the LMO definition from the Cartagena Protocol is a cornerstone to our approach. According to it, a genetically modified organism is that possesses a novel combination of genetic material obtained to the use of recombinant DNA techniques or direct injection of nucleic acids into cells, overcoming natural barriers. So we use this definition to discriminate line by line, product by product, if we are facing a GMO or not. The main question here in this very simple decision tree is there is a new combination of genetic material. If there is no new combination of genetic material, the product is not a GMO. So the applicant is notified about it, and then he must deal with the regulator for conventional products. Using this decision tree, we have been able to take already some decisions regarding several products from new breeding techniques, including genome editing in particular. In this map, you can see 2 dimensions. One of them relates to the concept of modern biotechnology in the Cartagena Protocol definition, and that's a vertical dimension. And then the horizontal dimension relates specifically to the concept of novel combination of genetic materials. So this map allows us to classify the most likely classification that products will have, although the classification is made case by case by analyzing what is exactly the change made in the genome. In here, you can compare the dynamics of regulatory decisions, beginning in 1996 with the first approval of a genetically modified live product for agriculture in Argentina. And then since 2016, the decisions regarding if certain products from new breeding techniques, including genome editing, are GMOs or not. So blue versus red, you can see that products from new breeding techniques, which is mostly genome editing, have been presented to the regulators with a dynamic that goes much faster than GMOs. And also you can spot there those stars that highlight the fact that there has been some products developed by genome editing or other new breeding technique that when presented to the regulators in Argentina, it was decided that those products are GMOs. This is another interesting feature of the caseload that we have until now. As I was saying, most of what we call new breeding techniques, the concept is not specific to Argentina. It has also been used in the European Union preliminary studies about how to regulate these technologies. I was saying, among the so-called new breeding techniques, most of the cases that we are receiving are genome-edited organisms. And these organisms, if classified according to the biological kingdom and the trait of the modification, we can see that there is a lot of diversity. I mean, in the 3 biological kingdoms, and many traits, not only for agricultural purposes, but also for consumer preferences, increased health, also increase animal welfare, many diverse traits of interest to different sectors of society. If we compare that for a moment with the caseload of genetically modified organisms that have been approved over a very long extended period, 30 years, we can quickly see a difference in the GMO universe over 3 decades. Mostly we have plants in commercial use and some vaccines. And for those plants, the prevalent traits are only 2 of interest to the farmers: herbicide tolerance and insect protection. Only now we have some other traits arising the horizon, like drought resistance that I mentioned earlier. But then if we compare to new breeding techniques, genome editing, we can see that there is much more diversity in what can be offered to society in a relatively short term. If we now look at the profile of the applicants in the left, you can see that in the last 5 years of taking decisions regarding products from new breeding techniques and genome editing, most of the applicants or developers are local Argentine companies and Argentine public researchers. I mean, research institutions. That's very interesting, because if we compare that to GMOs, that's a minority in the GMO universe, as it happens in most other countries around the world. And then the other half of presentations received for genome-edited products. Most of course, all the other half are foreign, but most of that part is coming from small companies, not from those multinationals that have been mentioned before. But this doesn't mean that multinationals are not interested. They are also taking stage, but for some reason, perhaps they are not so fast in adapting or making use of new opportunities. We are seeing that most of the applicants and developers are from small companies or public research sector. In Argentina, as I was saying, we are seeing how Argentine scientists are developing products using CRISPR and other genome editing tools. Also, startup companies that were created solely to develop genome edited organisms for agriculture, or established companies, important seed companies in Argentina that now have created a biotechnology lab to have their own biotech crops using genome editing. And this is not restricted to plants. The same thing is happening in animals, both the public sector and the Argentine biotech companies that, for instance, work on assisted reproduction and cloning, now also have projects using genome editing. And this is because we are having this policy not only in Argentina, but in neighbor countries, as I'm going to describe next. In Argentina, we also, or the government also have work on the aspect of public communication. So this is made not at the back of the public, but public is participating and knows about these new policies. And I'm about to finish now. Argentina also is working with other countries, trying to find ways to harmonize and have the more rational approach for the regulation on genome-edited crops and animals for food and agriculture. Finally, the same thing that I have described for Argentina is happening in other Latin American countries. In only 5 years, we have 8 countries in our region that have a very similar approach to the regulation of genome editing for agriculture. And of those, 3 of them, Chile, Brazil, and Colombia, are already receiving applications. And alongside with the governmental decisions, we can see that their public research and private biotech national sector are working on developing products of local interest. So with this, I finish. Thank you, Reimagine Europa, and all participants for the opportunity to share this with you.